Okay, so by now you've heard of the different vaccines that are in circulation in the United States. There are several major trials that are going on in the United States, and the vaccines that have been developed by Pfizer and BioNTech, they are now in use. They have been granted emergency use authorization as of December 11th by the Food and Drug Administration. And this emergency use has been approved for people ages 16 and older. So a question that people may have is, people are calling these novel vaccines, or rather the experts are calling these novel vaccines as opposed to traditional vaccines. Traditional vaccines are vaccines that expose the body to proteins that are made by viruses and bacteria, whereas these novel vaccines are DNA and RNA vaccines. And more specifically, the vaccines that have been granted emergency use are RNA vaccines. And RNA vaccines are considered safer than DNA vaccines because it is harder for RNA vaccines to get integrated into the cell's genome. Essentially, what all of these agents do, whether it's DNA, RNA, or protein, is they trigger the body to generate antibodies against some agent that you want the body to create antibodies against, right? So in this case, this agent is the protein on the surface of the virus, which is called the spike. And this spike is what gives coronavirus the name corona. So it's like a spike on top of the virus. And this is what results in attachment of the virus to cells. And the spike protein, what it does is it enables the virus to attack cells, that is attached to cells, and then infect the cells. So what you're essentially doing is by using a protein vaccine, which is the traditional vaccine, or the novel RNA vaccines, you are enabling the body to create these antibodies against the spike protein that the coronavirus has. And by doing that, you will essentially make the people immune because the people will now create these antibodies against the specific virus. And the other thing about this is whether it's a DNA or RNA vaccine, they essentially work in using the same mechanism. But if it is the RNA vaccine, it is closer to what you want it to do. So DNA is first transcribed to RNA, and RNA is translated to protein. So if you are using DNA or RNA vaccines, it is easier to inject rather than the protein vaccines. Again, DNA vaccines, they can get more easily integrated into the genome. RNA vaccines less so. DNA vaccines also have to be injected using special pulses like electrical pulses. So looks like RNA vaccines are the way to go, but we are early in this new technology being applied to millions of people. And as the vaccines are being injected to these people, we will know very soon what is the efficacy of these antibodies that are generated by these people as a result of injection of these RNA vaccines. So here is what you need to remember. The novel vaccines that are being injected to people are RNA vaccines, more specifically mRNA vaccines. And these mRNA vaccines will result in the generation of antibodies against the spike proteins of the coronavirus. So hopefully this was a little bit helpful in understanding the terminology and technology that is going into vaccinating millions of people and making our world an immune place to these bad viruses.